Biochemistry, at its core, studies the chemical processes within and related to living organisms. It is a laboratory-based science that brings together biology and chemistry by using chemical knowledge and techniques. Understanding biochemistry is fundamental to comprehending biological structures and functions, exploring how they relate to health, disease, and innovative therapies. One of the primary components of biochemistry is the study of biomolecules such as proteins, lipids, carbohydrates, and nucleic acids, which play critical roles in cell structure and function. Proteins, composed of amino acids, perform a vast array of functions including catalyzing metabolic reactions, DNA replication, and transport. Lipids are crucial for forming cell membranes and energy storage. Carbohydrates provide energy and are involved in cellular communication, while nucleic acids, such as DNA and RNA, store and transmit genetic information. Enzymes, another key concept, are catalysts that speed up chemical reactions without being consumed in the process. They are highly specific and operate under optimal conditions of pH and temperature, with their activity often dependent on the presence of cofactors and coenzymes. Biochemical pathways, composed of a series of chemical reactions, are essential for managing the complex tasks cells must perform. These pathways ensure precise and efficient processing of metabolites for homeostasis and energy. Metabolism, which includes catabolic and anabolic pathways, is the sum of these biochemical reactions, converting nutrients into energy and building blocks for cells. The importance of biochemical pathways. Biochemical pathways are pivotal in maintaining cell function and responding to environmental changes. They provide insights into cellular function and regulation, highlighting how cells harness and convert energy, respond to stimuli, and maintain their internal environment. These pathways illuminate how biomolecules interact within the cell, allowing us to understand disease mechanisms and developmental processes. The glycolytic pathway, for instance, is central to cellular energy production, breaking down glucose into pyruvate with the net gain of ATP. It illustrates cellular respiration's complexity and interdependence with other metabolic pathways. In contrast, the photosynthesis pathway in plants converts solar energy into chemical energy, sustaining life by providing oxygen and organic compounds. The study of these pathways enables researchers to identify points where interventions may correct metabolic dysfunctions. For example, the Krebs cycle and oxidative phosphorylation pathways' role in cellular energy supply underpin research into metabolic diseases and potential treatments. Emerging technologies, such as metabolomics and systems biology, further our understanding of these networks. They rely on comprehensive biochemical pathway analysis, enabling us to interpret complex biological data holistically. This approach not only uncovers the intricacies of metabolic regulation but also paves the way for personalized medicine and advanced biotechnological applications. Historical milestones in biochemistry. Biochemistry has evolved significantly since its inception, with numerous historical milestones marking its development. The field emerged in the 19th century as scientists bridged discoveries in organic chemistry and complex biological processes. Friedrich Wöhler's synthesis of urea from inorganic compounds in 1828 challenged the prevailing vitalism notion, suggesting organic compounds could be synthesized outside living organisms, and laid the groundwork for biochemistry. The isolation and crystallization of enzymes by Eduard Buchner in 1897 was another significant milestone. Buchner's work led to the understanding that enzymes could promote biochemical reactions outside living cells, a revelation that earned him the Nobel Prize in 1907. This discovery positioned enzymes at the heart of biochemical research and pharmaceutical development. In the mid-20th century, biochemistry underwent a paradigm shift with the discovery of DNA's structure by James Watson and Francis Crick in 1953. This breakthrough elucidated how genetic information is stored and transferred, catalyzing advances in molecular biology and genetic engineering. The elucidation of the double helix structure of DNA has since influenced countless studies in gene therapy, forensics, and biotechnology. Further advancements were achieved with the emergence of recombinant DNA technology in the 1970s, which revolutionized genetic research by allowing scientists to manipulate DNA sequences and express foreign genes in host organisms. This technology facilitated the development of modern biotech products, including insulin and other therapeutic proteins. In summary, the foundations of biochemistry are built upon fundamental concepts and historical advances. The field continues to expand, driven by technological innovations and a deepening understanding of biochemical pathways, impacting numerous areas of science and medicine.